listening to the Pharmacy Podcast Network.
In a world where one-size-fits-all medications dominate the pharmaceutical industry, precision medicine brings a ray of hope for those seeking customized healthcare. Pharmacists have a unique opportunity to help people in need of specialized testing to ensure medications work as intended. Welcome to PGX for Pharmacists, where we unravel the wonders of precision medicine and its potential to revolutionize the way we approach pharmacy care. Get ready to uncover the secrets behind pharmacogenomics and how it's transforming lives one genome at a time.
Hello, everyone. I'm your host, Dr. Becky Winslow, and you're listening to the PGX for Pharmacists podcast that Whelp magazine recognized in 2021 as the 9th most listened to genetics podcast in the world. On the PGX for Pharmacists podcast, we explore all things pharmacogenomics related, and our mission is to educate and advocate for PGX. We accomplish this mission through exclusive interviews with highly qualified and well-experienced pharmacogenomics industry leaders, such as today's special guest.
And my name is Banas Sarami, the co-host of PGX for Pharmacists podcast, a pharmacogenomics medical science liaison, and a mentor to pharmacists. Connect with us on LinkedIn and let's get a conversation going. We want to hear from you and how you're impacting pharmacogenomic stakeholders and what you have learned throughout your journey.
Okay, so without any further ado, I'm extremely pleased to introduce to our audience Dr. Thierry Dervieux, the Chief Scientific Officer at Prometheus Laboratories. And Prometheus Laboratories is a reference clinical laboratory that's focused on the diagnosis, prognosis, and monitoring of immune-mediated inflammatory diseases. So thank you, Dr. Dervieux, for joining us on the podcast today. I'm excited to share your and Prometheus's story with our audience. In particular, I'm excited about you sharing your career journey as a PharmD, PhD, and Chief Scientific Officer and designer of the Predictor PK ADA, which is a precision-guided dosing test for the optimization of Humira, Remicade, and their biosimilars. So one of Banaz's and my main goals for this episode of the PGX for Pharmacists podcast is to expand our audience's notion of what a PGX test looks like and to inspire them to think bigger than the traditional box PGX test most of you are familiar with. So, Dr. Dervieux, I'd like to start the podcast by having our guests introduce themselves and elaborate on how you are a pharmacogenomics expert.
Yeah, thank you, Becky, for having me. So I am a pharmacist with a PharmD with a doctorate in pharmacokinetics. I completed my studies in France and I came as a postdoc fellow to work in the United States about 20 years ago, to work on the pharmacogenomic of anti-cancer agents, primarily 6-mercaptopurine as well as methotrexate. After my postdoc, I moved in industry for Prometheus. So I have a large experience in the implementation of pharmacogenetic testing in immune-mediated inflammatory disease. Our lab, Prometheus, was the first clinical laboratory in the United States to offer the thiopurine, methyltransfer genotyping, as well as the thiopurine metabolites. So I have over 70 publications in the field, and I'm very excited to be on the postcard with you today.
All right, so thank you for qualifying yourself as an expert. So let's jump right in and delve into your current PGX work. So if you'll tell us a little about Prometheus, specifically, what is Prometheus's mission and how are you guys going about accomplishing your mission?
Yeah, sure. So Prometheus is a reference clinical laboratory based in Southern California, in San Diego. The company has been there for over 25 years. We are specialized in the differential diagnosis of autoimmune GI disorders, gastrointestinal disorder, on inflammatory bowel disease. And over the years, we have developed a portfolio of differentiated solutions to facilitate the diagnosis, the prognosis, the monitoring, as well as therapy selection with pharmacogenetic testing, which we are offering through our clinical laboratory. And most importantly, recently, we are developing some testing solution with the predictor PK test to optimize treatment to biologics.
Okay, well, that's great. Can you also tell us about the Prometheus library of precision medicine tests for inflammatory bowel disease for patients, how they benefit medication therapy management stakeholders across the IBD's patient's journey from diagnosis to treatment to disease monitoring to remission, and how they differ from other lab tests for IBD and its treatments?
Yes, so our clinical laboratory offers some highly specialized tests to facilitate the diagnostic of to facilitate the differential diagnosis of inflammatory bowel disease. So we are offering a testing solution with serological testing, for example, Pianka as well as microbial antibodies that are present in Crohn's disease, as well as other autoantibodies that are present in ulcerative colitis. These are conditions that are somewhat difficult to treat, and we are offering those tests to help gastroenterologists, first of all, to establish a differential diagnosis of IBD as compared to other conditions, typically irritable bowel syndrome, as well as other gastrointestinal disorder. When the diagnostic is established, we offer testing to establish a prognosis, where we're going to inform the clinician that the patient has a more aggressive disease that will require more aggressive treatment, where we can provide a testing solution to initiate the most appropriate therapy for the patient, with a testing where we are basically determining some genotyping with the thiopurine methyltransferase genotyping, for example, where we can indicate that the patient is likely to present with a side effect to those medications. And once the treatment is initiated, we have a portfolio of solutions to facilitate the monitoring of the disease, of the inflammatory bowel disease, as well as the dosing optimization with the answer test, which measure blood level for monoclonal antibodies that are indicated in the treatment of IBD, starting with infliximab, adalimumab, as well as ustekinumab and vedolizumab. So we have a comprehensive portfolio to surround the clinician with a variety of testing solutions, with our goal being to improve the outcome of patients with IBD. And I think that the pharmacist has a very important role to play from that perspective.
So Thierry, could you elaborate for us more on the predictor test, especially since you designed that test? We'd really like to know, what did that take and what role does it play in your suite of testing?
Yeah, sure. So the predictor test is utilized when the patient is receiving treatment. It's been specifically designed to optimize biological disease modifiers such as infliximab, adalimumab, that are core therapies in the treatment of inflammatory bowel disease, as well as other immune mediated inflammatory disease. What the test does is you collect a blood specimen, with dosing information, and what we do is to provide guidance to clinicians with respect of the best dose to give in order to achieve the level which is the most consistent with the disease control that needs to be achieved for the patient. Typically, a vast majority, about a third of patients tend to be unresponsive to these very expensive medication, not because they don't have typically because they have pharmacokinetic, suboptimal pharmacokinetic that makes them unresponsive because not enough drug has been given. So what we do with the predictor test is to basically estimate the pharmacokinetic parameter for the patient, and from then report the best dose to give in order to achieve the level which is consistent with the most effective disease control to be achieved for the patient. So we have developed the test for the infliximab as well as adalimumab, which is Humira. These are anti-tumor necrosis factor, and we are also developing the test for vedolizumab as well as ustekinumab that are widely used also in the treatment of inflammatory bowel disease.
Wow. Prometheus's suite of tests goes well beyond the PGX testing that our audience is most familiar with, which typically only includes SNPs for CYP genes and some pharmacodynamic genes. This is really exciting. Genes and biomarkers related to immunology are not commonly found in what I call the boxed PGX tests, such as those made by large laboratory manufacturing companies, where the panel has a set number of genes, and it was developed by a larger laboratory for maybe smaller laboratories' use. So my understanding, having talked with you extensively, Thierry, is that immunology has fewer PGX tests available because it's actually more difficult, say, than oncology to research and develop tests. So could you elaborate for our audience on the difficulties that are associated with immunology research and developing tests for immunology versus, say, oncology?
Yeah, sure. So in immunology as compared to oncology, there is no such a thing such as a somatic mutation. Where, for example, you're going to have a BRAF, with that indicates that the patient, is likely to benefit or not from some treatment. In immunology, this is far more complicated for a variety of reasons, starting with the fact that, the response to these medication are multifactorial, and the fact that the mutation that are the single nucleotide polymorphism in the germline, which can potentially associate with outcome have low penetrance, meaning that they're going to have a weak association with response to those medications. So there is a necessity in immunology to combine multiple genetic polymorphism together in order to achieve some performances characteristics that will make the clinician order the test, and most importantly the payer to pay for the test. So this field has been is moving forward. There are some tests that are being developed right now, but the biggest challenge is to be able to achieve again the threshold of performance that makes the test is valuable enough to being again ordered by the clinician and utilized to the benefit of the patient.
I couldn't agree with you more. I've worked on the payer side or market access side of pharmacogenomics, and even with a box test for which there's a lot of research data available, even with those, it's sometimes difficult to get payers to see the value. So I absolutely agree with you. The fact that you guys are investing in producing the data necessary says a lot about your laboratory, and how committed you are to this testing and how you believe in the testing. So I just want to make sure that our audience recognizes that, Prometheus doesn't simply provide tests to determine if drugs for IBD will be effective and safe. And maybe what the dose of the drug should be for the patient, but you have that whole suite of tests. The diagnostic test for the differential diagnosis, all the way through remission. So can you elaborate? You elaborated on it some in the previous question, but can you tell us the difference between how you had to actually develop the tests you didn't, in other words, purchase a test from another manufacturer with the biomarkers that you include in your testing. Can you elaborate on how much more difficult it is to develop a test from scratch?
Yeah, sure. I mean, this is challenging for multiple reasons. First of all, you need to have a clinical data set available with specimen available, informed consent obviously available. So we are leveraging at Prometheus a large biobank of specimen. As I said, Prometheus has been founded 25 years ago. So over the past 2 decades, we have been able to assemble a large substrate of data and specimen, which we are using to establish our proof of concept, if you will. And then when we have identified some genetic polymorphism that are adequately associated with disease outcome and disease progression, as well as toxicity, then we are entering validation phase where we are using validation cohorts where we are again combining multiple modalities together, patient demographic as well as genetic marker together with serological marker actually, to come up with some multivariate models that are again bringing the performances characteristics of the pharmacogenomic test or its combination with other marker to the level where it's supposed to be in the first place to meet payer satisfaction and obviously again patient to the benefit of the patient and to improve its outcome, the outcome.
I think what you're describing really is the future of pharmacogenomics. In other words, not singling out pharmacogenomics as the end all and be all in the treatment paradigm, but using a PGX test in combination with, like you mentioned, other serological tests, maybe other genetic tests. But I think what we want our audience to really wrap their heads around is that PGX is just a piece of that larger puzzle, from diagnosis to treatment to remission. So I think you guys are you're already in the future. In other words, you're already providing all these different tests, like you mentioned, to facilitate from diagnosis to remission.
That's correct. Yep.
So you've given us so much great information about the tests that you guys offer. Can you explain to our audience your newest test, the responder test, and what role it will play in the paradigm from the diagnosis of IBD to remission?
Yes, sure. So we are doing things a little bit different than other. We do believe that it has to be simple. You can obviously construct some very, very complex algorithm, and there are some tests that do that with some fairly sophisticated machine learning based tools that are available using neural networks, those sorts of things. But we have taken a different approach where with the responder test, where basically we are taking an approach which is very simple to address the first and foremost, most important aspect of predicting response to medication is the pharmacokinetics. You cannot be responding to a drug if the drug is not given, and you obviously cannot respond to a drug if the drug is not metabolized adequately. And this is what we are doing with the responder test. We are addressing some fundamental issues with those biologics, for example, the anti-tumor necrosis factors such as infliximab and adalimumab. It is well known that those drugs, first of all, are prone to immunization, meaning that the drug itself is recognized by the immune system and digested by the antigen-presenting cells, if you will, where you're going to have an immune response mounted against the drug to produce immunogenicity that will severely impact its pharmacokinetics, where the levels will be inadequate to produce the desired anti-inflammatory effects. So we are, with the responder test, we are combining two things together. First of all is the genetic test itself, which predicts the risk of immunization. The name of the test is an HLA DQ-A105 allele that promotes the presentation of infliximab, for example, to the T-cell repertoire in order to promote the clonal expansion and the formation of the anti-drug antibodies, together with another dimension, which is the clearance, which is as important.
Yes.
One of the key issues with the monoclonal antibodies such as infliximab or adalimumab are in fact any monoclonal antibodies that those drugs are cleared and consumed from the central compartment, if you will, since we are doing a little bit of pharmacokinetics here. And if the patient presents with a high degree of inflammatory burden, is going to have the patient will have a high clearance, and that's going to worsen in the presence again of the HLA-DQ-A105 genetic marker that associate with immunization. So our test is a combination of both. These are the predictive factor of pharmacokinetic origin, which we combine together, where the patient presenting with a risk of immunization, as well as accelerated clearance due to the fact that the patient has high inflammation, or due to the fact that there are some intrinsic pharmacokinetic properties that makes that the patient, will clear the drug very, very fast. For example, due to the inefficient recirculation of the drug itself through in the reticuloendothelial system. Together, those patients presenting with these poor prognostic factor of pharmacokinetic origin will tend to be severely underdosed, will not be responding to the drug adequately, and they probably should in the first place if you are able to address the exposure. So what we do with this test, we will be able to inform the clinician that the patient is at risk of achieving suboptimal pharmacokinetics, and therefore being able to adjust the dose to start with more adequately, so that the proper exposure is achieved during induction to achieve a better outcome. And I think the pharmacist will have a very important role to play here.
Absolutely. That information is priceless in the management of these medications. So thanks for elaborating on that.
And if I may add, in our previous conversation before the recording of podcasts, we had discussed you guys' robust platform for collaborating with payers to obtain market access and reimbursements for the test. But without stealing the thunder from Prometheus market access and reimbursement team, can you please briefly detail how Prometheus has proactively worked with payers to solve the problem, the population health problem, by building the evidence payers want to see about your test before you go to the market, and then build the test and then hope the payers will see the value and the result, and then that will improve the market access and reimbursement for your precision medicine test.
Yeah, so briefly, I'm probably not the right person to answer that question. We have a very, very efficient market access group at Prometheus that does a splendid job. But what I can tell you that we have an evidence development plan in place where we are establishing the clinical utility of our testing solution by demonstrating payer value with respect of patient management and the impact of our technology on physician behavior. We have already commercialized two of those tests for which we have initiated the Empower studies that already provide differentiated and value to the payer, where the clinicians are basically using our technology to make treatment decision, as well as some prospective clinical utility study which we are initiating to again demonstrate the payer value. We can certainly follow up with you can certainly follow up with our market access group as appropriate there. They can fill you with more information.
No, that totally makes sense. That totally makes sense. But we're excited that you're also a PharmD. So how did you get to this role of outside the box path? There may be pharmacist student or pharmacist wanting to switch or transition into a role such as yours, which is a chief scientific officer, and want to learn more. So how would you, can you talk a little bit about that?
Well, we are a clinical laboratory, so in order to be in my role, you need to have expertise in clinical laboratory science. So for the students, it's basically, to do the PharmD degree and then complete the PharmD degree with a doctorate, which is focused on clinical laboratory science. So you can achieve, all the elements you need to be, for example, board certified as medical laboratory director. So you can, this is a great opportunity, I think, for pharmacists. There is an absolute need to have the clinical pharmacists provide drug information to healthcare professionals, as well as assist patients with the monitoring of their disease, the effectiveness of the therapy, and monitoring the side effect and the toxicity from those medications.
Well, Thierry, I know our audience is going to have additional questions for you. I mean, you've provided them with so much great information, but it's only the beginning of what they could possibly learn about the testing that you do for IBD and even your career path. So if you wouldn't mind telling us, because we have to wrap up, unfortunately, this episode of the podcast. Could you tell us how our audience members might be able to contact you directly?
Yeah, I can be contacted on my email at tdervieux@prometheuslab.com.
All right. Well, thank you again so much for joining us on this episode. We really, really hope that our listeners' ideas of not only what PGX can be, but how PGX can be utilized in a comprehensive testing suite. We really hope that our audience will listen in and learn this information. And to our audience, thank you for tuning in. We really hope that you've learned from this episode. We do a whole lot of PGXing here on this podcast. We talk about PGX science, clinical application, and the business of PGX. So we'd love to hear from you. Love to hear from you. What can we teach you? What more can we teach you through our podcast? So please drop us a message on LinkedIn and let us know.
And please share this link to this podcast link episode with everyone so they can tune in and listen to the PGX for Pharmacists podcast. Leave us a review on Apple Podcasts or Spotify. And you can also visit us on PGX4rx.com to listen to all our other episodes.
Well, thank you.
Thanks for your interest in PGX and for spending some time with us. Please share this podcast and leave us a review on Apple Podcasts or Spotify. For all of our episodes, please visit pgx4rx.com. That's pgx4rx.com.